So welcome everyone. My name is Lukas. Thanks for having the chance to present here. The title is Targeted DIA on Orbitrap Analyzers using the software that we developed, Spectronaut. And it's quite an open title, so I will take the liberty and discuss quite a lot of different things. So first, introduction. So I wanted to give some, maybe that's interesting for you, a quick history on diagnosis and data independent acquisition, how this all started for us and how we got involved in that. So as was already said, we used to be working in the lab of Rudy Eberswald before, and at the time when we just made the transition where Biognosis was founded, and when I started to work for Biognosis, these ideas around DIA and target analysis, they started. So, but before that, a whole lot of different DIA methods were already published. So in 2004, for instance, the term DIA was coined, as far as I know, was a publication of Venable et al. After that, in 2006, there was a first publication about MSE. It's the DIA flavor of waters, you could say. Then in 2008, Biognosis was founded here at the ETH as a spin-off, as a startup company. In 2011, we already made the first measurements on a Q-Executive. It was a demo instrument in Bremen where we acquired some DIA data because we thought back then we will only buy this instrument, it can also acquire this kind of data. And since there was a quadrupole in front of the Orbitrap, it was really the first time that you were able to acquire such kind of data. We also started developing Spectronaut exactly at this time point. Then in 2012, there was the first publication about target analysis of DIA data, the publication from Chile, from the Eberswald lab. In 2012, we published a paper on IRT, normalized retention time. Back then, we were mainly using it for MRM, but later on, it proved to be very useful also for the analysis of DIA data, and I'm going to show a few slides about that as well. Then in 2012, we made the first public release of the Spectronaut software. 2013, there was this multiplexed flavor, again a different way of acquiring the data, so a different acquisition method. They also used targeted analysis of this data, and then in 2015, maybe also worth mentioning, the first time that, or it was not necessarily the first time, but the first time it was really shown to work very well, that you would analyze your data not with a spectral library, but more a bit like you would analyze shotgun data. It's this DIA Umpire paper. So now an introduction about proteome quantitation. So usually, if you want to do quantitative work in proteomics, you have a number of conditions. You already saw the very nice presentation from Olga. I'm not going to say a lot about the statistics, how you analyze such kind of data. But in principle, you are interested in different conditions and you want to quantitate proteins. And if it's DIA, usually it's on a whole proteome or sub-proteome basis, so you want to quantify a lot of different proteins. And so the goals are, they can be manifold, but you might be interested in mechanical or in the mechanistics of biological processes. You might also want to do more systems biology, things like comparison or clustering of proteomes or comparisons to transcriptomes and so forth. Or you might want to do biomarker discovery, very classical. And then maybe interesting for you also the kind of samples. I listed here samples that are mostly interesting in a commercial setting. So the kind of samples we analyze as services at Biognosis, it's actually mostly human. It's very often blood or urine or CSF. Sometimes you also have tissue, sometimes cell lines or secretomes, we also did already. And then very seldom you also have model organisms model organisms are actually very seldom. We also have plants quite a lot, that might also be interesting for you. And then the number of samples that we do for these DIA type of experiments, they are in the range of 6 to hundreds of samples. We also had more than 100 samples, also projects with more than 100 samples. The conditions, again, can be manifold, disease states, drug treatments, time courses, or whatever you can think about. And then the main LCMS technologies, you can use labeled workflows, so like SciLag or TMT, or you can also do label-free, and here I'm going to focus on label-free only. So all what I'm going to show is label-free quantitation. So a quick comparison, I think you already saw this in the DIA introduction yesterday, but a quick comparison on a DDA, a shotgun method, and a DIA method. So the big difference is that in DDA, you have the semi-stochastic sampling of precursors during a run, so the instrument decides which precursors are being sequenced. Whereas in the DIA method, the method is very reproducible. You always measure exactly the same things, and you're just going to see what is visible in the scans that you acquire. So it's very reproducible. The method is very reproducible. Then there's also a difference in terms of quantitation. At least, let's say, if you quantitate on MS1 for shotgun, and if you quantitate on MS2 for DIA, if you do that, it's remarkable that roughly 25% of the time is used to acquire scans that are used for quantitation in shotgun, whereas in a DIA method, roughly 90% of the time is used to acquire scans that are later on used for quantitation. So this already tells you that it's probably quite a strong quantitative approach. And then this is a small, that's actually you could say it's small technicality, but it's also the title of this course, so it's targeted analysis. So you can analyze, shotgun data is analyzed starting from your raw data. So you start from the spectra and you query your search space. Whereas in DIA, at least if you use a targeted analysis strategy, you start from your search space, your spectral library, for instance, and you query your data with your spectral library. And that's just, this means that all the algorithms that you are used to processing the data, they are different, and you can also use different kinds of algorithms, and many of them are quite elegant. So this is an advantage to analyze your data in a targeted manner. So target analysis using spectral libraries, you do not necessarily need to use a spectral library in the common sense, but that's what is usually done. So this was first published in this publication from Gillet et al. And you can use it to generate extracted ion currents. You can do this on MS1 as well as on MS2, and actually Spectronaut does both. So it does generate extracted ion currents on both levels of spectra. And if you visualize that, it looks like as the following. You have a spectrum, you have your ions that you are looking for, and you follow them over time, and you get these typical elution profiles from the chromatography. This then looks like MRAM data, and it can also be analyzed in a similar way. And I'm going to have a couple of slides about that later on, but we borrowed the tools that we developed for MRM to analyze this kind of data. That's the mProfit algorithm. So here are just a couple of figures from this mProfit publication and how you can use this to automatically identify peaks and also to estimate false discovery rates, which is very important with these large data sets. So spectral libraries, how they are usually generated, usually with shotgun proteomics. On the right, you can see what happens if you shoot the same sample again and again. So you can see that you reach some sort of saturation. So you can use that to generate spectral libraries, for instance, and that's what we have used mostly in our laboratory. So we didn't do any fractionation in most of the experiments. We just shoot the sample many times, and this we get a quite large spectral library, much larger than what you see in a single shot in shotgun. So the minimally required information is the precursor ammo set and the fragment ammo sets, but it's very helpful if you have additional information in the spectral library, such as the index retention time, the IRT, how we call it. It's just a description of where your peptide is going to elute in the chromatography. So it means that you have measured this peptide before, which you did in the shotgun run. Then it's also very beneficial to have the relative fragment ion intensities. There have been lots of studies that compared the fragmentation of different instruments and so forth, and generally you can say that nowadays most of the instruments can generate fragment ion spectra whose relative intensities are very comparable. So it's you can very well determine your relative fragment ion intensities once and reuse them, and it works very well. Optional, you can also have the peptide sequence and other information in the spectral library, but that's then less important for the quality of the data later on. Just quickly, I want to introduce a term that we use at Biognosis. It's basically how we term our workflow, HRMMS, and there are a couple of ingredients there, so we always use this HRM kit. The IRT kit is an ingredient of the HRM kit. Then we use spectral libraries, like has been published by Gillet et al. And we acquire the data on a QExactive, so we have a QExactive in our laboratory, and we analyze the data with Spectronaut using targeted analysis. And quickly some words about our software, just in very general. So these are two software packages. One is for the analysis of DIA data, Spectronaut, and the other one is for the analysis of MRAM or PRM data. It's called SpectraDive. This one has now also newly been available to academia for free, so everyone can download the software as well. And for Spectronaut, we have to mention that our business model is that you require the HRM kit, so you have to buy the HRM kit and then you can use the software for free if you are from academia. And so the software is easy to install, provides high quality results. So in the very beginning, when we started with all that, we of course were interested how would DIA perform compared to DDA or to shotgun. Because you can do shotgun, you can do MS1 alignment, and if you do everything very well, it can provide reasonably good results. You have this missing value issue, but we really wanted to do a benchmark to see what is really the better approach. So we started this project back quite a long while ago. And for this study, we generated a number of very well-defined samples. So we generated 8 samples. They all had the same background matrix, a HEC293 cell line extract. So a very complicated background matrix. And then we spiked in proteins with known absolute amounts and with a large dynamic range and also with small and large fold changes. I'm not going into details here, but you can see the spiking schema that we chose here. So we had 3 different sets of spike-ins, and this was an exponential dilution, so we covered a really large dynamic range, and so we had all the fold changes in there. And then to make it really fair, we also used the acquisition sequence where we basically were always acquiring HRAM, also DA and shotgun, DA, shotgun, so that it was really the same setup, same chromatography, and we really also followed kind of best practices for shotgun. Then we had a look at the CVs. So we used the MS1 quantities for shotgun. We processed the data with MaxQuant. And we used the MS2 quantities. There we always used the sum of fragment ion intensities for a precursor. This is our quantity that we used. And we looked at the CVs first. And what we could see is that the CVs from the DIA, they were lower. That was already very promising. But then, so the second part, we also wanted to show. Actually, it's not on the slides, if I think about it, but so the second part we also show in the paper is we also wanted to look at the fold changes and which method could perform better in finding regulated proteins, for instance, if you would use MSTATs. And we also performed this analysis in collaboration with Olga Witek. And what we saw is that also there, DIA was better in finding changed or regulated proteins. And you could say that DIA was better in terms of precision, the CVs, and also in accuracy, so finding the fold changes of proteins. And what was also very striking, or what you could say is probably the reason for this result, was that when you looked at the reproducibility of peptide identifications, this is how it looked for shotgun if you look at the 24 replicates. So what is depicted here is the full profiles. This means peptides that were identified in all the runs when going from 1 to 24. And you can see that this number drops very dramatically. So a lot of peptides are not identified in all the 24 runs. Whereas if you look in DIA, most of the peptides are identified and quantified in all the 24 runs. So this was very striking. And if we just randomly sample 100 precursors, so there's no bias, we just took 100 precursors that were identified and we look at the DDA data and at the DIA data, this is how the raw data looks like. One has to say we didn't use MS1 alignment for this publication because we didn't want to use it neither for shotgun nor for DIA, but we did the experiment ourselves. It improved the shotgun results a little bit, but not dramatically, so it didn't change the message of the whole analysis. And also you could also do DIA alignment, or you could also do alignment for DIA actually, which we didn't want to do for this publication because it would have blurred the message of the publication. So also in the course of this work, we were also looking at how can you make good DIA methods. And of course, we wanted to generate good methods on an Orbitrap instrument. And what you can see here, so is a peak depicted basically in the dimensions you have in the data. So you have the M/Z dimension and you have the time dimension. And usually we just display the time dimension and the MZ dimension is encoded in the color, basically. And what is important is that you balance the number of data points per peak so that you have, so there is an optimal number of data points per peak. It has an influence on many other parameters in the method, but if you keep this number of data points per peak stable, that's very beneficial. Then there's other things to consider, so these are the parameters you can use to counterbalance. For instance, the resolution, the field time is connected to that. Then also that you have enough ion flux for the spectra, so you need enough ions for the spectra, you also need to consider that. Then, of course, you can change the number of precursor segments that you use in your method. You can change the precursor range. And you can change this for MS1 and MS2 independently. So there's a lot of things you can change. So now a few words about these data points per peak. So if you consider a peak capacity in your chromatography, so in your LC, of 400, let's say you have a peak capacity of 400, that's quite reasonable nowadays. It's can even be rather low, but it's decent. So let's say we want to have 7 to 8 data points per peak for quantitation. You could design a method that's just a dummy method. So you would do 1 MS1 scan and 6 MS2 scans, and this would fulfill the requirement to have enough data points per peak. Now let's say you in your laboratory, you have a different chromatography setup and you have a peak capacity of 700. Then this would mean you couldn't just take my method, because you would end up with too few data points per peak. So you will have to adapt your method. You can do this in many different ways. Here I just depicted a change where you would use less precursor windows. So here instead I used 3 precursor windows, and you would end up again with your roughly 7 data points per peak. So it's very important, if you benchmark methods, that you keep the number of data points per peak stable, because otherwise you might end up drawing the wrong conclusions. So because the results you see, they are an effect of the different number of data points per peak instead of the other parameters that you changed. So that's something that we found is very important. Then another thing that was enabled in 2014 by Thermo, of course it makes a lot of sense to accumulate your ions while you are scanning. And before this wasn't possible on the Q Executive, but in the since the beginning of 2014 you can do that, and it also improved the results quite dramatically. We were also interested in the influence of the chromatography on DIA, so whether during this time it has been published that with increasing peak capacity for shotgun you would increase the number of peptide IDs, and we were able to do this on a Q Executive Plus in the lab of Paolo Picotti. So we had 2 setups, a peak capacity of roughly 300, one with roughly 600. This corresponds to this setup with a really long column, 50 centimeters, very small beads. So of course high back pressure, you need a UHPLC for that. And we acquired triplicates, DIA and shotgun again, 2-hour runs, and we looked at the performance of these 2 methods. So here is the details of the 2 methods. We had to change the DIA method because the peak capacity increased, the peaks were narrower. So we went from 19 segments to 9 segments. And the results were as follows. So what we basically saw is that when you go from shotgun to shotgun, 30 to 50 centimeters column, this is the increase you can get on the number of protein IDs, and this is what you can get for DIA. So we saw a similar effect for both methods. The effect is a little bit smaller here because we couldn't use a large spectral library here. So if you would lose a large spectral library, it would probably go up a little bit more. So the effects are very similar. Also, if you look on peptide level on the right here. So if we go from here to here or go from here to here, the effects are very similar. So this was our conclusion. Peak capacity influenced both methods very similarly. Okay, now a few words about Spectronaut software. So very quick, Spectronaut is very fast. I think it's the fastest software available at the moment. It can be a few minutes per run, and I'm not talking about very small runs. So you can, on a Q Executive HF, you can have runs that have 10 gigabytes of size. So it can analyze those runs very fast. It can also analyze very large experiments. Hundreds of runs. We have done this in our lab for customers, and we also have users who do such large experiments. You can very easily generate spectral libraries, either from MaxQuant or Proteome Discoverer. We use the concept of IRT. You need the HRAM kit spiked in, as I said before. We have this automatic peak picking, FDR estimation, so you don't need to manually go into the data and pick these tens of thousand peaks manually. You can do it all automatic. We have an automatic interference correction. I have a slide about that later on. We also have label-free normalization incorporated. And we also have automatic quality control. So because you have to spike in the HRAM kit, we can do quality control automatically. So you automatically have readouts about the LC and the mass spec without you doing anything. You can just have a look at it after you analyzed your experiment. And we can also do differential abundance of proteins, so t-tests. And yeah, that was it. So in quick, these are the features of Spectronaut. Spectronaut 7 has been released at the beginning of the year. And so now about a few words about specific features, so about this normalized retention time. I think you already have seen a presentation about IRT, but I hope it's not too repetitive, but I still want to show you this very quickly. So IRT, so let's talk about the way forward. So you would have two standard peptides, this one and this one, the red ones, and you define a value for those two peptides, and you measure on a linear gradient a third peptide, this one here. And if you have assigned an IRT for this one, and you have a linear gradient, you can also calculate an IRT for the peptide in the middle. So for this one, it's a simple linear equation. You can get an IRT for a third peptide by fixing just two fixed points. You can also imagine the Celsius scale. It was fixed at 0 and 100, which is water that is ice and boiling water. Just 2 points were fixed. It's the same concept, basically. And then you can also go the way back. So once you have your IRT values and you have measured the two fixed points on a new chromatographic setup, you can predict the retention time of this peptide independent of the gradient or gradient length or the gradient used or whatever. So it goes in both directions. And we also defined this set of 11 IRT peptides. Two of those peptides, this one here and this one here, these were the fixed points. It was defined as 0 and 100. Now you could ask, why didn't we take the first one? We didn't take the first one because it showed more fluctuation than the second one, because usually how the gradients are set up, in the very beginning, you have more fluctuation. That's why we chose the second one. And so these are 11 non-naturally occurring stable peptides. They're evenly spread over RT range, and two peptides were used as fixed points. And now how can you use that? In the beginning, we used it a lot for scheduled MRM, because if you schedule, you can measure more peptides. And you can use the same concept for PRM, of course. But you can also use it for targeted extraction for DIA data. And so this is what we started to use it right afterwards and implemented it into Spectronaut. So yeah, so let's imagine you generate a spectral library, let's say with Spectronaut. If you have your IRT peptides in your sample, you can determine IRTs for all the tens of thousands peptides you have measured in shotgun. And Spectronaut can do this automatically. So if you generate a spectral library, all the peptides measured have an IRT assigned to them. And now what happens if you measure all these peptides again on a gradient and you plot the RT versus IRT? If you have a linear gradient, basically it shows you just a straight line. If you use a nonlinear gradient, it then looks like that. So you can basically predict the retention times of all the peptides in your gradient. And you can also see if in your LC, for instance, something is crooked. If you have a hiccup somewhere, you can immediately see it in this visualization. Now how do you use that for targeted analysis? So yeah, it's very simple. If you have your full extracted ion chromatogram, you can just zoom into your full extracted ion chromatogram and actually only look in the region where you expect peptide. And this would be the zoom in here. So you only look in this region and you do the peak detection only in this region. Of course, you choose the window in a way that you would never miss a peptide, or let's say almost never miss a peptide. Then you can also check what happens if you have no IRT in your spectral library, or what happens if you have IRT, or what happens if you have very precise IRT in your library. And this is what we have done for a poster at the ASMS here in 2014. And we looked just at the identifications you get. You can see that you get more IDs if you have a very precise IRT, but also that your identifications get more reproducible. So the same peptides are more reproducibly identified. The better your IRTs are, in general, the better your spectral library is. So that's something you would expect. Okay, now a few words about FDR estimation. My first presentation I gave here at the SRM course was only about that. And I think it was, people didn't find it so interesting, so I kept it to a relative minimum here. So the basic approach is, as I said, it's borrowed from MRM data, so we developed this initially for MRM data. And the idea is that you would simulate truly absent peptide signals. It's also done in shotgun proteomics with your reversed FASTA file. And after that, you can use the method of story to estimate Q values. And these are a number of figures from this MRM publication, mProfit here. And what you can see here is the score distributions for targets and decoys, so for true and false signals in this case, for the subscores that we developed for mProfit. So here are 8 subscores. In black you can see the distribution of the decoys, and in gray you can see the distribution of the signals we knew that they were true. And you can also look at this at the final discriminant score. This is a combination of all the scores here. So you can see here it's a bit different. You can see the true signals here. So these are the present peptides, and these here are the absent peptides. And the red ones are the decoys. They simulate the absent peptides, basically. And so we use this concept also to check the FDR estimate of Spectronaut in a little bit different way. So the idea was we wanted to see whether the decoys are really truly, so they are a fair representation of absent peptides, because, I quickly go back, if this red distribution would be completely off, for instance, if it would be over here, your FDR estimates would be terribly wrong. So you want to check that this distribution here overlaps with the gray distribution below here, and that's what we did. So we wanted to simulate truly absent peptides, and for this we took assays from an organism different to the one analyzed. So we analyzed human sample. We mixed the spectral library with assays from E. coli. So this depicted here. So we made a spectral library for human. We made a spectral library for E. coli. We mixed the 2 spectral libraries. We only took high abundant peptides here, so only the ones we were certain to be able to see. We analyzed the human sample with Spectronaut with the mixed spectral library. And the result looked like that, so as you would expect, or how it's supposed to be. So the E. coli assays we couldn't detect. This is the gray distribution here. These are the human assays. We could detect them all, basically. And the decoys, they are perfectly overlapping with the E. coli assays, also depicted by a QQ plot. And we also knew the FDR, so we could calculate a known FDR for each cutoff, and we could also estimate it with our method that's depicted here. And the take-home message is, if you make a cutoff at a certain point, our estimated FDR was always higher than the true one. So we had a conservative estimate on the FDR. This is what we wanted, and this is also what you would expect from the from this paper of Story. It's a conservative estimate of the FDR. Okay, that's all about the FDR. Spectronaut can also do interference correction. So here's an example of a precursor, 2 fragment ions, light blue and dark blue. They show an interference, so there's another peptide that has roughly similar precursor and fragment mass, and it shows an interference, basically. But what you can use is you can determine a consensus profile of those fragment ions here, and you can compare all the elution profiles to this consensus profile, and if you detect an outlier, you can actually remove it from quantitation. And this is implemented in Spectronaut. If you enable the interference correction, it will consistently remove some fragment ions that show interferences in some of the runs. And it will do this, of course, consistent across all the runs, such that quantitation is okay in the end. And if you enable this option, you can see that the CVs go down for both median and average CVs, so you gain a few % of precision, which is nice. Okay, then we also have implemented a profiling algorithm in Spectronaut. You could say this is the analog to the MS1 alignment in Shotgun. And the idea was, we have presented this at the poster in 2013. So the idea was that you would find the region of elution of peptide that is below the limit of detection. So this would be the limit of detection. Of course, yeah, this is just visualizing it. So it means that you couldn't identify this peptide with an FDR of 1%, but you can see that there is still some remaining signal, and it would actually be nice to use this for imputing, as Olga mentioned before. It would be nice to use this signal to impute it into the data matrix, because it will just lead to better results. And the idea was that we would use the surrounding signals as well. So for instance, this interference signal we could use as a landmark, and if our algorithm is able to find these neighboring landmarks, it should also be able to find this region here. And we implemented this using cross correlations. And yeah, so the results are on the next slide. So we check this again with proteins that is spiked in known amounts, and therefore we could check the precision as well as the accuracy of quantitation with this profiling enabled or disabled. And yeah, so we saw that it improves both. It improves the precision as well as the accuracy a little bit, again, a few percent. So depicted here, so if profiling is enabled, that's the orange one, it is more close to the profile that we knew we spiked in than if we wouldn't have used profiling, which is the white one. Okay, so now I have a few slides on benchmarking studies with it, so benchmarking with these typical HeLa hex cell lines on a new type of instrument, Q-Executive HF, and also some slides about complete proteomes and why this is interesting, or is at least interesting to spend some thoughts on that. So the Q-Executive HF has an ultra-high field Orbitrap in. It's almost the same as a Q-Executive. The biggest difference is the Orbitrap, which has a higher resolution. Basically, it's on the next slide. So you have almost double the resolution at identical scan time, or almost double the scan rate at the same resolution. And yeah, of course, this opens up a lot of possibilities for better DIA methods. One example is depicted here, so you can use double the number of precursor windows now, which of course will increase your selectivity if you do that. And yeah, so we went on the Q-Executive, we were able to measure in Bremen on a Q-Executive HF in a collaboration with Thermo. And the goal was basically to identify as many proteins as possible in 1 or 2 hour runs. I know many people find this a bit boring, but it's actually important for us, and because it just basically improves the general data quality that we deliver to customers, or you can also cut down on your time that you use for measuring. In the end, that's important. So we generated a spectral library from 3 different cell lines, HEC, HeLa, and Churkat. And we didn't use any fractionation here. We just had decently long runs of 3 hours, replicates, which led to a spectral library of above 7,000 proteins, 122,000 peptide precursors. And we designed 2 methods for a 2-hour gradient and a 1-hour gradient. And we acquired DIA runs of a HeLa sample in triplicates again and analyzed it with Spectronaut 7. And these were the results, so just the number of identifications. So basically, we saw really impressive numbers. That's for one. So there was a big difference between the HF and the Q-Executive Plus. And the hallmarks of the data looked very similar. So it was reproducible, and quantitation was very well. So with single shots, we could cover almost, depending on the number that you use for what is the expected expressed proteomes in these human cell lines, between 45 and 65% of the expected expressed proteome. Then we also looked at the CVs. It's very important. It's a quality feature that we always report to our customer. The median CVs, you can see that for 1-hour runs, it was a little bit higher. The interesting thing here is that if you look at it in this way, so this is thousands of peptides and the corresponding CVs for the 1-hour run and 2-hour run, you can see that most of the peptides have CVs below 50%. So you get a very large number of precursors between 30 and 40,000 or even 50,000 for the 2-hour method that have really low CVs. There are only a few outliers. So the data quality is generally very high, even if it's label-free quantitation. So this was our take-home message. Then one very striking thing was that in the 1-hour DIA runs we acquired, we identified 45,000 different peptide precursors, corresponding to roughly 5,500 proteins or protein groups. And when you now take a shotgun run, like best practice shotgun run, 1 hour on the Q-Executive HF, you will acquire no more than 36,000 MS2 spectra. So that's very striking. So this was nice to see for us, but also a bit unexpected to see it so clearly. But basically, you can identify more peptides in a DIA run than you can do MS2 spectra best practice on the same instrument. And how can you explain that? So, of course, shotgun is a very sequential method. You have to acquire one MS2 spectrum after another. Whereas in DIA, it's a very parallel method. So you will measure many peptides in parallel. This is how you can improve the coverage or the throughput of your method. So the parallel identification instead of the sequential identification really enables a leap forward in amounts of peptides quantified, reproducibility, and also in quality of the data. Okay, then we wanted to push the limits a little bit. We went 3-hour runs. We measured the 3 different human cell lines now quantitatively in triplicates. It's all single shot, so it's really just 3-hour runs. So these are 9 3-hour runs. And we also enabled now the DIA alignment that I explained you before, so this profiling algorithm. And when doing that, we ended up with almost 8,000 protein groups. And this was only 1 week of measurement, so we didn't have so much time to really optimize everything in Bremen. So we were really impressed by this data. CVs were in the range of 10%, median CVs, and we identified roughly 85,000 precursors. So you can get very high protein coverage in single shots, and this also means that you can measure many samples, so you can get a very comprehensive data sets in a small amount of time. Depending on the number of expected proteins, again, this is 55 to 80% of the expected expressed proteome, so it's very impressive. So yeah, I have this slide still in, it's from other presentation. I think it's interesting because, if you spend a few thoughts on that, there is a couple of interesting things. So why is coverage interesting, or why is it important? Why do you want to have a complete proteome? And there is a number of non-linear effects, so meaning with increasing coverage, these effects are not linear, they are overlinear, so they grow like that. So for instance, so this one is a linear one, the first one, but you don't want to have missing proteins in your data, and this is something we often have with customers. They want to do a DIA experiment, but they also have their favorite proteins, and now if you have a chance of 10% of finding those, it's not so nice, but if you have an 80% chance of finding them, it's much better. So that makes a big difference for them. Also, if you want to make comparative studies, for instance, between samples or tissues, the higher your coverage gets, the higher to 100%, the more likely you get a nice comparison, as depicted down here. So if you have 2 times 50%, you get 2 data points out of this hypothetical example. If you double your coverage, you get 100% of information out. So you see it's a nonlinear effect. That's why it's nice to get a high protein coverage, maybe in the range of 50 to 80%. Yeah, there's a number of other things. I'm not going through them right now. Yeah, then complete protein coverage, a little bit of literature, literature research I did here. So 10,000 proteins in a single cell line in 2012, then almost 12,000 proteins in 2014, but this was a liver, so different cell lines from liver. And then in 2015, this was a meta-analysis of 19,000 LC-MS runs. It's also the data included from these Nature publications. So they corrected their number down to roughly 15,000 proteins if you treat the protein error rate correctly. So this is the protein coverage if you do extensive fractionation. So maybe you can get close to these numbers in the near future with single shots without fractionation, which is interesting to us. Okay, then yeah, a little history of our laboratories. In the very beginning with QExecutive, normal HPLC, no ultra high pressure. We were in the range of 3,000 to 4,000 proteins. QExecutive with UHPLC, we have now a UHPLC, we get in this range here, and on the QExecutive HF, we were in this range. So it's continuously growing, it's interesting. Okay, then I thought this might also be interesting to you, 2 customer projects that we did. So these are projects that we sold to customers. They are blinded a little bit because we cannot show the data, but one project was about identifying proteins that differentiate cancer patients and their response to a drug. So we had 2 groups, class 1 and class 2. They respond differently to a drug. Also when they have cancer, they respond differently to a drug. And the idea was to find biomarkers for these 2 groups of patients. And we used our HM approach for that. The results were, so one quality control result was that if you do unsupervised clustering of the data, depicted here, so this is unsupervised clustering of the 2 patient groups, they perfectly clustered together. So that's very nice to see, of course. So basically by clustering, we could separate the 2 groups. This means, well, this shows you that the quality of the data was very high. But secondly, also they could find many biomarkers which they already thought were responsible for the 2, or that they were differently expressed between the 2 groups. And this was also interesting to see. We were also very quick in delivering the results. This is because we don't do any fractionation, so we can deliver the results very fast, within less than 10 days. A second example, I know this is just an example biomarker we found. Yeah, this is how you can depict the data in DIA. So this was a project in plant tissues. We had 5 different plant tissues. Again, we used our HRM approach, and they wanted to find proteins that are overexpressed in one of the tissues, and here's just an example displayed. So it also works with different tissues. Yeah, then a little excursion for the end. I'm not sure how much time I have left. Yeah, I think I have some time left. So you can also extend the approach of the target analysis to different kinds of data. For instance, to MS1 only data, and this is what we wanted to try here. So the idea was that you would only acquire MS1 data, and given a sufficiently high resolution, maybe you could also analyze this data in that way. And we used the normal approach, Spectral Library, and query the data or target your Spectral Library in the MS1 data. There are a number of potential advantages. It's a simpler workflow, also would be potentially a simpler instrument. All the effort is spent on spectra that are used for quantitation and identification and quantitation. That's interesting. There are, of course, a number of disadvantages. You need very high resolution. What do you do with isobaric species and so forth? Potential applications. You can do label-free quantitation, of course, but you could also imagine labeled workflows with very high plex if you want. And yeah, in principle, you can replace label-free shotgun. Of course, you need it for library generation, at least in the near future. So a comparison of the methods. So a normal DIA method, you make MS1 and MS2. Here, for MS1 only, you would only make MS1 spectra. And yeah, this has been published before, although not with a targeted analysis. So the Smith Lab has a lot of publications going back to 2000, actually. So already 15 years ago, they did such approaches on a FT-ICR instrument. They started from the raw data, so the processing was more shotgun-like, so you would look for your features in the data and so forth, process it to a single mass in the end, and then query your possible peptides if you have the processed features in the end. Yeah, they did it on FT-ICR instrumentation mostly, and it was mostly applied to non-complex proteomes, so simple organisms, or lower organisms like yeast, for example. So we had to do a couple of modifications of Spectronaut. The Spectral Library looks different, of course. We had to get a more robust calibration routine into Spectronaut. We also had to generate different kinds of decoys. So we also checked the decoys using the approach of this poster. And yeah, so after that, we went again to so we also did this in the lab of Paolo Picotti on the Q-Executive Plus. We had a UHPLC, 2-hour gradients. Here you can see the method that we used. We used 2 MS1 segments in this case. And we targeted the MS1 data with Spectral Library. And one important detail that is not on here. So what we did to get very accurate retention time, we made alternating DDA and DIA runs. So we acquired DDA, DIA, DDA, DIA, and so forth to get very accurate retention time for our first try. And it worked really well in the first try. So we could basically identify the full spectral library. You can also expect that because you have seen this all in the DDA run, so it must be there. But it was really striking how reproducible the data was. And also the CVs were very low, so very good. Here you can see the high retention time accuracy depicted, so it corresponded to a window of roughly 1 minute in a 120 minutes gradient. This was the accuracy we got in terms of retention time. So it was very encouraging. The resolution we used was 280K, so very high. This is the max you can do on a Q-Executive Plus. Then we went on to Fusion. There the maximum you can do is 450,000 resolution. We use that, it's a 1-second transient. Again, we use the alternating acquisition sequence, DDA, MS1, DDA, MS1. And then we wanted to check one very important thing, because we wanted to see whether you can decrease your retention time prediction accuracy and still get the analysis going, because normally you would like to store your spectral library for some time, and maybe you change your setup a little bit, and then the retention times get a little bit less accurate. So we wanted to see how robust we could make it on the Fusion to retention time fluctuations. And this is what we did here with a simulation. So we simulated spectral libraries that had inaccurate retention times when going from left to right here. And we checked the number of identifications we get. Here it's normalized between fusion and QExactive+. And when you can see with the higher resolution of the fusion, we got much more robust to fluctuations in the retention time. And actually, so this is standard deviation 6. It's what we got here. So you can see that it's quite a spread already. So this is very realistic, accurate retention times. It's actually even a bit pessimistic, I would say. And it was still working. So this was very interesting for us. So the higher the mass accuracy gets, the more robust you get to the retention time fluctuations. Yeah, that was already it. So in summary, Spectronaut is powerful software for target analysis of DIA and SWAF data. We have 2 major annual releases. Yeah, you can really say that DIA solves the missing value problem of shotgun. So you really get very few missing values, and that makes all the downstream analysis much more easy, much more simple. DIA label-free quantitation is precise and accurate. And yeah, the Orbitrap instruments, so the QExactive family and the Fusion family, they are very well suited for DIA because they have a quadrupole in the front. And yeah, it's also scalable to many samples. So you can measure hundreds of samples. I would say the limit is only the lifetime of your column, because as soon as you have to switch columns, the data quality decreases quite a bit. So with this, I would like to thank especially Diagnosis, Oliver Bernhard, he's the main developer of Spectronaut, all the others, and our collaborators at Thermo, UX1. Then Olga Wittek, she was involved, and Linyang Cheng, she was involved in the publication at MCP. They did all the statistical analysis of the data. And then at ETH, Paul Bersma and Paolo Picotti. So, and thank you very much for your attention.
Thanks a lot, Lukas. So, other questions?
Maybe I can start with one. Sounds super cool. What's not working?
Oh, it's not working. Yeah, you have to be a bit more specific. So,
what's the next challenge for Spectronaut developer?
What is the challenge for Spectronaut? Well, at the moment, for instance, what we don't do at the moment is this DIA umpire approach, so that you wouldn't need a spectral library, but you would do basically a database search of your DIA data. You can, in principle, do it also. You can just feed in a spectral library that is derived from a FASTA file, because Spectronaut doesn't need any IRT or anything else. You can just feed it the precursor MSET and fragment MSET. And we have also tried that, I have to say. So it works, it just doesn't work as well as with a spectral library. So that's why we stick to a spectral library at the moment.
I have a questions to the numbers. So you said the protein groups. So how did you define this? So is this only proteins from prototypic peptides, or?
Yeah, so for these benchmarks, we always use the same, excuse me, we always use the same number, so we basically take the protein grouping of max quant. So it's not only PTPs, it's all the peptides.
What is your limit of quantitation? Did you look into this?
So yeah, so for this publication in MCP, we knew the spiked in protein amounts. So we had these 11 proteins that we spiked in, and the lowest one was in the range of, in the attomolar range, and we could still detect it. So a very low limit of detection. And I have to say, this is the old setup we used. So now we have a UHPLC. This makes it more sensitive. If you would go to a QExactive HF, it would make it again more sensitive. So, but of course, when you talk about LOD, it will always depend a lot on the peptide you choose, on the response factor of the peptide you choose. If it's proteins, then it's a bit averaged out, or it's the maximum of the protein, basically. But yeah, so it's for this one protein, it was attomolar range.
Regarding the libraries, so you generate the libraries in a kind of sample-specific way, right? Your library matches very well the samples you're going to inject in DIA mode. Have you tried using larger-scale libraries? And something we talked about earlier was the effect on the false discovery rate at the protein level and so on when the target prevalence is lower. So is this something that you guys have thought about, or have you tried some big libraries? Is it supported in Spectronaut?
Yeah, so we did a lot of things in this direction. So in the very beginning, we wanted to see, so we made this comparison. What if we take a sample, make 6 fractions, for instance, SAX, and make a spectrum library, and then we take the same sample, but don't fractionate, acquire 6 technical replicates, make a spectral library. What will give me the best results when I do DA without fractionation afterwards? And what we saw, roughly 2 years ago, I would say, or 1.5 years ago, that it works better if you don't fractionate. But so I have to say, in the recent times, especially if you go to the better instruments, you can see that there is a certain tolerance to bigger spectral libraries. So with improved data quality and with improved software quality as well, you can have larger spectral libraries and you can have lower library recoveries, what we call it, and it still works very well. So in the beginning, we couldn't have used the pan-human library, for instance, to analyze our data. Now we can.
But do you actually compute the error rates at assay peptide protein level, or do you always filter everything from the assay level?
No, so we only calculate FDRs for precursor level. The reason is in DIA, because your search space is relatively limited, and the number of proteins you identify from the search space is very high, the amplification from precursor to protein is much smaller as compared to
for the small sample-specific libraries, right? But imagine if somebody wanted to use it to do this pan-human library extraction, then you would maybe have a problem.
Then it's a different story again, yeah.
So Lukas, you say that you prefer the technical replicate injection. So you have your sample of interest, and then you first do several shotgun of every sample. And you showed this increase on one of the slides, and it went up to 24 samples. So what's your typical technical replicate injection that you would do when you start a project?
Typically we do 6.
6 per sample?
Yeah.
And if you work with...
no, 6 per experiment or project.
Mm-hmm.
So, but of course, if you have a project where you have biological replicates, for instance, it makes a lot of sense to take 6 different biological replicates. Will also give you better results in the end. Or if you cannot, if you have very different biological replicates, then you can also make pools and measure 6 pools, for instance.
Okay.
But I have to say, so regarding this fractionation, so we actually run now the version 2 of these experiments, comparing fractionation to not fractionation, and I'm very curious what the result's going to be. We haven't analyzed it yet, but might be that now the conclusion is a different one. I don't know. So. But yeah, maybe to add, I wouldn't do 24 fractions, for instance. That would be probably too much.
Hi. For instance, for example, when you compare, let's say, two groups of samples, let's say control and disease, you have 6 biological replicates to each case. Okay, how do you define if a protein is identified? For instance, if you have it in 4 biological replicates in disease and 2 in controls, what do you say about that? Is it or not detected?
So, how we do it at the moment, you have 2 options, let's say. Actually, we use both options. So one option is you only use the data points that you get. And as we use a very simple model, we build ratios and we make t-tests of the ratios. So we just take all the ratios we can get and make a t-test out of it. So usually, because the reproducibility is very high in DIA, in the experiments, how we perform them, we do not have an issue with missing values usually. So we can do that. Or the other thing you can do is what I would call the DIA alignment or profiling approach. You can use these DIA alignment algorithms or the profiling algorithms, trying to recover signals that are below the detection limit or that you couldn't detect, and you just take all the values. So if you have identified a peptide once in your experiment, you will take all the values you get. So you will end up with a complete matrix, and you can consider the values that were below the FDR, you can consider them imputing. So this is the other option you have, and both of those options work decently well. So I have to say it depends a little bit on the type of project you have, which one works better.
I have a question regarding how you design the experiment, because you say your run takes up to 3 or 4 hours, yeah, and you have 6 replicate per experiment, replicate, and let's consider that we have 3 sample, biological sample different. 6 replicate, 3 hours. It means that you will have the same injection. It will take 12 hours for 1 sample. Yeah. From the first one and 12, I've noticed that on my experiments, I have decrease of intensities. How do you manage this?
Yeah, I think that's probably not something, excuse me, that is so easy to answer, because of course the longer your experiment is, the more difficult it gets to acquire high quality data over the days of acquisition. So it's not so easy. And I mean, you can try to design everything in a way that you can keep up the sensitivity over time. And I mean, there are so many different factors that influence that. For instance, if your sample is a little bit dirty and it has an influence on your column, you could add a pre-column, and then it would probably make your system a little bit more stable, but also more complicated. So, and you would really have to try to find out why your sensitivity drops. Usually it has to do with the chromatography rather than with the mass spectrometer, but it can also be the mass spectrometer that has sensitivity drop. And one other thing that we do, for instance, is we clean the instrument every 3 months. So the quadrupole, we clean it every 3 months, and the other parts we clean more frequently. So we have regular schedules of cleaning. We have quality control samples that we measure all the time, and we can see when the instrument is in a bad shape. So these are all the things that you need to do to be able to acquire a lot of samples such that the quality stays high. And then there's actually one other thing you can do. So you can make the acquisition sequence in a way that it will be less, that the statistical analysis, or let's say that you can still do a statistical analysis afterwards. So you can basically do replicate blocking. So this was something that was suggested by Olga Witek when we made this publication. So you can have the same replicates of the different conditions in the same block and randomize within those blocks, and then have these different blocks. If then your instrument has a little bit of a decrease in sensitivity, it will not influence your statistical analysis. So of course, it's very bad if you measure your first condition and then your second condition, and your sensitivity drops, then you will find many regulated proteins which are downregulated in the second condition. But of course, that was only a technical reason, so you should by all means avoid that.
Yeah. And I've noticed also the samples also degrade if it stayed too long time in the dual plates also. It sometimes influences the results.
Okay, so
even if they are frozen in 5 degrees, I saw the
sensitivity drops over time. Yeah, that's something we can probably also depends. Sorry. So probably also depends on your samples or on your, I don't know, auto sampler, but usually we cannot see that the sensitivity, or that the sample degrades, let's say within a week or within half a week. Usually we don't see that. So I will also be at the dinner tonight, so if someone has more questions, you can also ask then.
Thanks a lot, Lukas. So we are perfectly in time. So the next lecture is the MStats lecture. It will be upstairs again, but we will start in 15 minutes, so you can shortly then go up.